Sign in. There is that. Okay, can somebody confirm you can hear me?
I can hear you.
Great, thank you. So today you are supposed to read through textbook chapter through the end of the section on compound light microscopy. You need to do that before beginning lab 3, as well as beginning lab 4. Lab 4 won't be until next week, but lab 3 will be this Thursday. Any question about that? Today, we're going to discuss and begin the infectious disease project, as well as discuss lab 2 on aseptic transfers. For the infectious disease project, scroll to lab week 2. And you can click on this link and get the infectious disease research project. It's a 100-point project and it is due May 18th. Let me open it up here. I wish it wasn't a funny color. All right, in the infectious disease project, it's a 100-point lab project. You're going to specifically focus on a specific infectious disease, determining the underlying cause, what microbe causes it, what the microbe does to the human body, how we treat the disease, the social, economic, and political factors affecting our ability to deal with the disease. And you're going to become an expert on one particular disease. That's the goal of this project. You need to pick an infectious disease and then check with your instructor about your disease. If no one has your disease and your instructor agrees to it, you may work on that disease, but you need to clear it with me first because everybody's going to have a different disease. And I want to make sure it's an infectious disease and it's narrowed or focused enough so you can actually write a decent report about it. For example, if you wanted to do something like pneumonia, I'm going to tell you no, there's too many different organisms that can cause pneumonia. You have to choose one organism that causes pneumonia and then write your report on that. Any question about any of that? Like somebody asked about writing the report on something caused by E. coli, and I told her, no, you can't do that because E. coli causes too many different diseases. You can pick one disease caused by E. coli and then write the report on that. I haven't heard from her, so I'm not sure, or I haven't checked my email today, so I'm not sure if that topic has been taken, but I don't know. Anyways, you're going to be learning about your assigned disease. And make sure you clear it with me because if it's not an infectious disease, I will not allow it. Like cancer is usually not infectious. Diabetes is not infectious. However, there are some infectious diseases which are not communicable, like botulism. Diseases like that are allowed. It is infectious, meaning that you can get it, but you're not likely to spread it to somebody else. And then you submit a written report. It must be typed, single-spaced, 1-inch margins, and it must have at least 12-point font. Smaller than 12-point font, I'll have trouble reading it, and so you'll get graded severely. So make sure it's 12-point font, so I have an easy time reading it. You must have a cover information. I do not want a cover page. Put your cover information at the top of your first page of text. The text page, the text body itself should not exceed 5 pages, single-sided pages. If it's more than 6 pages or under 4 pages, you will get a penalty. So it's supposed to be around 5 pages of text. By the text, we're talking about your written stuff. I'm not counting the bibliography or the references. I'm not counting pictures when I'm counting the pages of text. You must describe all aspects of the disease, and that should be in the report content. The report content will be worth 60 points. There's an additional 15 points for grammar, spelling, flow, and readability. As much as possible, the text should be written in your own words. Any information that is not commonly known. Oh, good, the lecture, Zoom, oh, not assignment, but the lecture Zoom page just finished. So my computer will speed up now. Any information that is not commonly known, either directly quoted or paraphrased, meaning you've written it in your own words, you must give credit for a proper source within the body of the text. Normally, it is at the end of the sentence. There is one exception. I'll talk about that exception. And you give that by using an in-text citation. So like when you're talking about how many people have died in the United States from COVID-19, that's not common knowledge. So you must give an in-text citation for where you got that number. If you're saying something like COVID-19 is a lethal disease and it's killed lots of people in the world or even in the United States, that's common knowledge. Notice the difference? If you don't use in-text citation and you just state something, that would be plagiarism. If you plagiarize 3 or more words from somebody else and don't put it in quotes and say where you got it from, you'll get a 0 on this project. So do not plagiarize. And by the way, this assignment will be graded for plagiarism, meaning when you turn it in, there's a, what do you call it? An app using Turnitin website, which will actually check your paper for plagiarism. And it's gotten so good, it actually is looking at similarities to other students' reports. And so what used to be anything above 20%, I would go look and see why the plagiarism score says there's a similarity to another paper of 20% or higher. It's gotten to the point where so many students have turned in the report on, let's say, whatever disease you're talking about, that it might be similar to the wording of another student. And that's just a problem that I ran into. And so what I've done to eliminate that is I've simply upped the, I'll look at it now if it's above a similarity score of 30% or higher. Anything less than 30%, I'm not even going to look at it. Usually, the 30% scores are from quotations that people have been given, and I can remove all of those and then take a look at it. And I'll look and see that the student is also changing the words. Like if they copy a paragraph from some other place and they've changed no words, you're going to get a 0 because you're plagiarizing from another source. However, if you go in there and you change several words, I will say, well, this student has made some changes, not very many, but at least they've changed some. And so I'll let that go. But that will give me a plagiarism score. It won't be 100%, but it will score that. Okay. Any questions about plagiarism? You must use an in-text citation whenever you're quoting someone or whenever you're given knowledge which is not common knowledge. How you do an in-text citation, it's going to be worth 10 points. Let me, sorry, this looks weird, so I'm having a hard time. I'm having a real hard time here getting a new one just because it looks so weird. There it is. So you're going along and you're typing your thing, and then you say something which is not common knowledge. Let's go wise. Then you should give an in-text citation. An in-text citation is normally the last name of the author in parentheses, followed by the year. If it's two authors, it will be similar. We're just going to give you an example. Oops. And then if it's more than two, you just give the first author and then say Jones et al. Period, comma, and then the year. This is about the only time you can use et al. In a citation, and that's in the in-text citation. And the complete references, which is not an in-text citation, that's at the end of your document. So the end of the document, you then go references, and then you give the full citation. So that would be Smith, usually abbreviations. You can give names. And then you give the year, and then the complete source for where you got the information. This will differ what is that called? I forgot what that's called. The address. So for a web page, it'll be something like, oh, that should be a comma there. It'll simply be the web address. And that would be something like www dot, let's go, I'm trying to think of a national journey of Academy of Sciences. I might not have that right. And then whatever the web page is, we're going to go HTML which is, it's going to be longer than that, but it's giving you an idea. That's for a web page. For a journal, the complete references, whoop, come on, will be the journal of the National Academy of Sciences. That's what I meant. Didn't have that right. Then you give the volume number. And then the page number. Is that right? Journal, volume number, page number. If it's a book, it's a little different. Let's go with that. I think it's chapter in the book. Title of book. Author of book. Which is actually editor, I guess. Publisher. And then city of publication. Any questions on any of that? So that's a complete reference. Now, when you're giving the complete references, you are not generally allowed to go Jones et al. We'll just go 2022, and then the complete source of the citation. You are not usually allowed to write et al. In the complete references. You must spell out all of the authors. There is one exception, and that is if there's more than 7 authors, meaning there's 8 or more, you must spell out 7 of the authors. Actually, it's the first 6, and you don't need to know this. Spell out the first 6 authors, then go et al., and then give the last author. Okay, that's the only time you can use et al. And for practical purposes, I would just tell you, don't ever use et al. In the complete references. And then you will be okay. Any questions about any of that?
I have a question about the reference.
Yeah?
So you said spell out first 6 authors. Is that both of the MLA and APA?
It's both of them, yes.
Okay. Thank
Like
you.
I said, it's just easier to spell them all out. The only time you can use et al. Is like when you have 8 or more authors. And that's just a rule. All right, where were we? Here we go. The sources you cite in your paper should be scientifically appropriate and reputable. This includes government information sites, USDA, FDA, scientific journals, popular magazines that are known to be reliable, like Scientific American, National Geographic, Time Magazine, Newsweek. The New York Times is also a publication which is credible. You may not cite non-credible sources like the National Enquirer. What was a kid's magazine that was humorous? Mad Libs, something like that. You may use books and textbooks on biology, microbiology. If you look at your textbook, you'll notice it's all reviewed. Any source that is reviewed would be acceptable, as well as a scientific, appropriate, and reputable source. A variety of scientific, reputable television shows, like from the History Channel, PBS, are acceptable. Do not use sources from religiously, politically, or privately funded publications or websites. Do not use a source from a personal website. College and university pages may be used if it is made by an instructor. However, if the site contains student work, do not use it. Wikipedia cannot be used because anyone can go in there and change what is on Wikipedia. So do not cite Wikipedia for a reference or an in-text citation. However, you may use Wikipedia to get started on your project, and then you can use the references in Wikipedia. Like when you want to cite something that's not in common knowledge, you can get the information from Wikipedia, but you can't use Wikipedia as an in-text citation or a full reference citation. All right, the literature citation page should have a list of all the sources cited within the text. So all of your in-text citations should be in the full references. The literature citation should be the last page of your assignment. You can use any citation style you want. You must have at least 4 unique sources referenced in your literature citations page, and they must be scientifically appropriate. And generally, I can just look at your references and determine whether they are appropriate or not. Complete literature citation page will be worth 15 points. So 10 points for the in-text citation and 15 points for the literature citation page. Many students do not use the correct format for either the in-text citation or the full references, in which case they get marked down. So make sure you have it as I stated. The final report must be submitted in the folder in Canvas. It must be submitted either as a doc, a Word doc, a docx document, MS Word, rich text format, or writing format. It will be checked for plagiarism. I do not accept PDF files because sometimes PDF files cannot be checked by Turnitin, and I want to be checking for plagiarism. So if you have another program, you need to save the document down as a rich text format or as a MS Word document and then submit it. Any questions about any of that?
Yes, I have a question about the how many total work reference you were asking? At least 4?
At least 4.
Okay, thank you.
reputable sources, yes.
Okay, so that's peer reviewed, right?
I'm sorry?
Um, it must be peer reviewed.
No, not entirely. A reputable source can include things like Time Magazine or New York Times, and those are not peer reviewed.
Okay, so as long as
As
reputable.
long as it's a reputable source.
Okay,
I don't know about the History Channel and PBS if they're peer reviewed. They probably are.
okay. Thank you so much.
All right, all scientific journals will be peer reviewed, all textbooks will be peer reviewed. Oftentimes, like Scientific American, is peer reviewed. And that would be a good way to make certain it's a reputable source if it's peer reviewed. All right, the project content of the text, use subject headings within your paper so I know where each section begins and where each section ends. You must have the causative agent. That's what causative agent is causing the disease. If it's a bacterium, a virus, a prion, or eukaryote, you need to give different information. For example, a bacterium, you have to talk about the characteristics of the cell, what its gram reaction is, what its cell shape is, what its growth arrangement is, any metabolic capabilities, anything that's unique about that disease. You're going to talk about different things if it's a virus. For history, how long have we known about this disease? Historically, how was this disease treated? What is the history of vaccine development? If your disease does not have a vaccine, all you need to do is state no vaccine has been developed. Okay? But you do need to state that for full credit. You can also, if you've done the research, you can state there's vaccines in development. And then describe the changes in our knowledge and attitudes towards the disease throughout history. Like the Black Plague, we've had a lot of changes in our knowledge and attitude towards this disease throughout history, because initially people did not know what was causing it. They did not know that microbes were causing it or how the disease spread. And it's true that people were quarantined, but literally out of fear, which probably was appropriate because it could kill people. But um, anyways, we now know that all you need to do is get the correct antibiotic treatment and you can be cured of the plague, the Black Plague. Even for COVID-19, we've had changes in our knowledge and attitudes. If you haven't noticed, if you go to the store, almost nobody is wearing a mask nowadays. When I'm at the store, I'm about the only one in the store that actually wears a mask. There usually is a few other, but not very many people. Changes in our attitude towards disease. And then talk about epidemiology, describe the prevalence and the transmission of this disease. Where in the world is it found? State if it's found all over the world. Like the flu, it's found all over the world. How many people are currently infected? If that's not known, state it's not known. For Ebola, for example, currently I think 0 people in the world, at least humans, have Ebola. The rate of new infections would then be 0 for that disease. What are the rates of the morbidity and mortality? Well, it depends on the disease. And what is the reservoir of the pathogen? Like, for example, the reservoir for malaria or other people and mosquitoes. What are the modes of transmission? For malaria, it's the bite of the Anopheles mosquito. Anopheles is a genus of mosquito. So not all mosquitoes can transmit malaria. Then talk about pathology. You can read about what must be in pathology. talk about response and treatment. You can read about what needs to be in response and treatment. Then talk about the social, political, and economic considerations. You can read about what's in that. I have a copy of the instructor reading grading rubric. It's found on the next page. Your assignment will be graded using this rubric. I'll be grading you for your cover page, and you know something, I'm going to change this right now. Change that to a 2. Yours won't have that, but I'll change it. And then let's change that down to a 9. So you get 2 points for the cover information. Info is at the top of the page of the first text of content. The causative agent is worth 9 points. Make sure you have a subheading labeled causative agent. The history, worth 10 points. Epidemiology, 10 points. Pathology, 10 points. Response and treatment, 10 points. Social and political economic issues, 9 points. Let me talk just a little bit about history, for example. So you must be, to get all 10 points, you must talk about how long we've known about this disease, the history of vaccine development and treatment, and then changes in our knowledge and attitude of this disease throughout history. If you talk about all 3 of these, you'll get 10 out of 10, assuming there's no errors. You'll get 15 points for the quality of your writing. If the grammar is good, the spelling is good, the punctuation, flow, and readability are all good, you get 15 out of 15. In-text citations, if they're correctly citing information that's not common knowledge and the in-text is in the correct format, you get 10 out of 10. The literature citation page, full references, in-text citations, they match the in-text citations, and there's at least 4 reputable sources, you get 15 out of 15. As I've stated, if you don't have the correct format, you will not get the full credit on the citations. That'll be a total of 100 points out of 100 points. Any question on this project?
I have one question just for reference. Is there a minimum page? There's a 5-page maximum. Do you have a minimum?
No, the maximum will be 6 pages. If you go over 6 pages of text, I will mark you down. If you go under 4 pages of text, I will mark you down.
Okay,
So the
thank you.
average should be around 5 pages, but anything between 4 and 6 will be good. That does not count pictures, does not count the references. I'm talking about the full references. Okay,
And that is single spaced, correct?
single space, correct.
Single space. You.
All right. Oh, I need to save that, and let me make a note. I need to upload that because I changed it. Grading. All right, let's talk about lab 2. Let's exercises. Lab Module 2, aseptic transfers. Upon successful completion of this lab, you should be able to 1, understand the purpose of using aseptic transfer techniques. 2, determine if aseptic transfer is used from the data provided. And then 3, define the following terms: ubiquitous, aseptic transfer, inoculation, and contaminant. In our local environments, microorganisms are ubiquitous. That means they're almost everywhere. This is problematic when working in a microbiology lab because we generally want to work with a pure culture, a culture containing one species. Louis Pasteur developed the technique for aseptic transfers to minimize the risk of culture contamination. We will learn how to make aseptic transfers in this lab. During inoculation, never lay the test tube cap or the Petri dish lid on the workbench, because if you do, the cap or the lid can pick up the bacteria, and then when you put the lid back on, you'll be moving the lid over the dish, and then the bacteria on the lid can fall down into the plate. To minimize contamination when inoculating media, you should tilt an end cap tube. The reason why, if you don't tilt it, the full surface is open. And if you tilt it like this, only a little bit of the surface is open. I can't do it here because I can do it a little bit. There's less space to drop down if you tilt it. So if you're tilting it, you're having less open for the microbe to fall down from the air. It's most likely going to fall here instead of falling in the open tube. So you tilt it to reduce airborne contamination. And when you're opening a Petri dish and you're inoculating the dish, you should have the lid partially covering the dish and then sticking the utensil that you're using to inoculating it inside and under the cover of the dish. Any questions about that? You should also work quickly, having the tube or the lid uncovered for as little time as you can. You shouldn't be talking. And obviously, if you feel you're going to sneeze or cough, you should stop what you're doing. Because obviously, if you sneeze into it or cough into it, you could contaminate it. That's why students should not be talking while they're working in the microbiology lab. You should sterilize the noops and the needles. That's a loop. I don't have a needle here. Before and after they're used. Before you enter, you should also sterilize your work area, disinfect the workstation, and then do that before you leave. So that if any organisms fall down on the workstation, they're removed. In order to inoculate transfer cells to new sterile media, we must make sure that we sterilize everything that can potentially contaminate the sterile media. And this is showing you some of the things you use to inoculate sterile media. First, you have a test tube for holding the liquids upright, a cap. You have something for sterilizing your loop. Back to incinerator, you stick it in when it's red hot. Make sure the wire portion of the loop is red hot before you pick up the bacteria and then move it into your sterile media. You should know the terms in this lab. Laboratory exercise 2. You're not going to be physically performing any of these activities because everything will be done online, but you will be watching videos of these activities. We expect that you will know and understand the material in these videos and in this lab. In procedure 1, you're going to transfer sterile liquid using a glass serological pipette. We have a little video demonstrating proper aseptic technique, so watch that in your lab. In procedure 1, you're going to use aseptic technique to transfer 1 ml of sterile water into a tube of nutrient broth. When you measure water in a pipette or in a birette, you'll note that the water will not go across straight. It'll form a meniscus like this. You do not read the meniscus at the top of the meniscus or the average of the meniscus. You read the meniscus at the bottom of the meniscus. And that's because glass and even plastic, the water will stick to it and then will form a meniscus. In procedure 1, we're going to look at the media and it will look like this. To begin with, sterile. And then use this table for interpreting your result and making your conclusions. When you write your result and your conclusions, the wording should be similar to the table. So if your nutrient broth is clear, that's your result, then the conclusion is no organisms are growing in the broth and aseptic transfer was successful. When you're stating your conclusion, you must talk about whether organisms are growing or not in the broth and whether the aseptic transfer was successful or not, meaning the wording must be similar to the table. If it's not, then you will not get full credit. You can use the same wording in the table, but it must be similar. That's for procedure 1. If the nutrient broth is turbid, hazy, or cloudy, organisms for procedure 1 are growing in the broth and aseptic transfer was unsuccessful. And then here's the results of procedure 1. Remember, the tube initially looked like this. So state your results for procedure 1 and then what the conclusions are. And that's right here. This will be for you as 0. Question 0, describe the initial clarity of the nutrient broth. State whether it's clear or turbid. And then in 1, describe the clarity of the nutrient broth after inoculating with sterile water and overnight incubation at 35 degrees Celsius. State whether it's clear or turbid. 1B, based on these results, what conclusion can be drawn? So state the conclusion there. And then 1C, list 3 sources of error with aseptic transfer technique that could explain why the nutrient broth showed growth of the microbes. Any question about any of that? You must give 3 sources of error. It must be a reasonable source of error, like a statement saying that the tube was initially cloudy would not be correct because we can see that the tube initially is clear. Any question about that? All right, that's procedure 1. Let's go on to procedure 2. Procedure 2 is going to be transferring a sterile paper disc using forceps into a tube of nutrient broth. And you can watch the video on that one too, but you can just remember it from procedure 1. So you have a nutrient 2 broth. You need to sterilize the forceps. In reality, we won't be sterilizing the forceps because the forceps are metal. And if you were to get them red hot, you then wouldn't be able to handle the forceps. What you do with the forceps is you clean them. They're not going to be perfectly sterile, but you'll remove most of the bacteria and microbes from it. And how you do that is you put them in alcohol and then put the alcohol in either the flame of the Bunsen burner or inside the Bechtel incinerator, and that will light the alcohol. And then you wait for the alcohol to burn off, and then you can cool the forceps, which that's not important for here, and then pick up the sterile paper disc with the forceps and put it into the tube of nutrient broth. And then incubate it. Once again, look at the table for the results for procedures 1 and 2. If the nutrient broth is clear, no organisms are growing in the broth, aseptic transfer was successful. If the nutrient broth is turbid, organisms are growing in the broth and aseptic transfer was unsuccessful. There's the results of procedure 2. So the P means procedure, by the way. There you can actually see the paper disc that we added, the bottom of the tube. And then answer the questions for number 2. For the results of procedure 2, describe the clarity of the nutrient broth after inoculating with sterile paper disc and overnight incubation at 35 degrees. State whether it's clear or turbid. And then based on these results, what conclusion can be drawn? Remember, you must give the similar wording from the wording in the tube. If you don't state whether organisms are growing in the tube and whether the transfer was successful or not, you will not get full credit. For procedure 3, we're going to be transferring Serratia marcescens to a broth. We have a little video for you to watch for procedures 3 and 4, demonstrating how to correctly transfer from a bacterial stock culture to sterile broth or a sterile agar slant. So in 3, we'll be transferring to a sterile broth. Some important notes about this. You only need to watch this video to 4 minutes and 23 seconds. In the video, they're using Bacterium E. coli, which is non-pigmented. We're using Serratia marcescens, which is pigmented. There's also an arrow in the video from 3 minutes and 5 seconds to 3 minutes and 20 seconds. Can you find what that arrow is? You can hear some sizzling, and that's because the instructor making the video made an error. In procedure 3, you're transferring aseptically Serratia marcescens to a tube of sterile broth. Serratia marcescens is pink or red pigmented. In a broth, it's pink pigmented. Broth cultures are problematic because it's difficult to determine if contamination has occurred. All the cells will mix together in a liquid broth. You will not see individual colonies in an individual broth. So if you had minor contamination, the tube will still look pink from Serratia marcescens. You would not know if you had contamination unless the contaminant were to be a significant, like the majority of the cells in the tube. So that's a problem with the liquid broth, and that is when you have slight contamination, you're not going to know about it. Here's the procedure for procedure 3. Use this table for the results of procedure 3. If nutrient broth is clear, no organisms are growing in the broth, and aseptic transfer was unsuccessful. If the nutrient broth is turbid, hazy, or cloudy, organisms are growing in the broth, and aseptic transfer was successful. Why do we want organisms to grow in the broth for procedure 3? Anyone? All right, you want organisms to grow in the broth because we're transferring organisms, Serratia marcescens, into the broth. So we expect organisms to grow in the broth. Here's the results of procedure 3. So answer the questions for 3. The result, describe the clarity of the nutrient broth after inoculating with Serratia marcescens and overnight incubation. Was it clear or turbid? Based on these results, what conclusions can be drawn? And 3C, how do you know whether you transfer the correct microbe, Serratia marcescens? All right, for procedure 4, you're going to be transferring Serratia marcescens to an auger slant. You watch the video on doing the auger slant. Serratia marcescens on a slant will be red pigmented. Contamination may more easily be detected than on a broth. When you're inoculating, you'll be inoculating either on a slant or a needle, and then you inoculate the slant, shown here, on a S-type motion. And if you have any growth growing, let me blow this up. Growing out of the slant, like something's growing right here, and it's not where you have the slant, then you know that's probably a contaminant. Another way to see a contaminant is Serratia marcescens should be red on this inoculation mark on the slant. And if you have a region right here which is not red, then you know you have a contaminant. Any question about any of that? So contamination can be more easily detected on a slant than in a broth. You can determine slight contamination on the slant. You cannot on a broth. Use this table for the interpretation of the results. For procedure 4, if the slant does not show confluent growth, no organisms are growing on the slant, and aseptic transfer was unsuccessful. If the slant shows confluent growth, organisms are growing on the slant, and aseptic transfer was successful. Here is the results of procedure 4. Then answer the questions for procedure 4. Is bacterial growth growing on the slant? All you need to say is a yes or no. How do you know? State how you know the bacteria growing on the slant. And 4C, does it appear to be the bacterium you inoculated or a contaminant? And you should state why, you know. All right. Any questions about put that in there. Any questions about this lab? If not, go ahead and start working on this lab. I will be here to 8 o'clock to help you with any questions on the lab. So if you have a question, ask before 8 o'clock or before the last student logs out. When the last student logs out, I'm going to log out as well. Go ahead and get started on the lab.